Hi. Now, before we start, I want you to imagine something. You know, a time when life first started appearing. Okay, so the evolution of life. It all started possibly. Okay, I'm not sure about it. Nobody's sure about it. There's a lot of theories. Let's just assume theory of evolution as we know it is true. So probably some amino acid aggregates started coming into picture, and your nuclear material came, the RNA, DNA, and then the nuclear membrane, and so that was the initial basic thing. Now organelles, all that, mitochondria, everything is coming into the picture. But for all this to happen, you need a compartment which is delineated from the water outside. So as we know it, life began in the primordial ocean. Okay, now in that primordial ocean, you have to compartmentalize. You need a separate space away from the ocean water. So that is your intracellular compartment. For that delineation to take place, you need some sort of a membrane. Okay, so that is where your plasma membrane first came into the picture. So now the question was, how is the cell going to do this? So there are different particles. Ultimately, you need something that is hydrophobic, something that is lipid soluble, hydrophobic, that can repel water, so that it can compartmentalize. Okay, now in this scenario, you can think of lipids. A simple lipid structure. Problem is it is hydrophobic. So ultimately, in water, if there's something hydrophobic, it tends to completely get expelled out. So you need some structure which is amphiphilic. Part of it is hydrophilic, part of it is hydrophobic. That is your phospholipid structure. So something like a phospholipid structure in a low concentration, what we observe is that you put it in a medium of water. Okay, at some point there will be a air fluid interface. So this thing rises up, goes, covers over on top, the hydrophilic part on water, the other part outside in air. So it comes out like that at low concentration. That won't help you because that is not compartmentalizing a separate thing called a cell. Now you increase the concentration a little more. What happens is a micelle is formed. So there is water all around. There is high concentration of phospholipid. What is it going to do? It is going to, the part of it which is hydrophilic is going to bind to the water, and the hydrophobic part is going to push inside. So all the hydrophilic part will be outside, hydrophobic part pushed inside, forming a micellar structure. So that part inside will be completely heavy. Now, how good is this? So again, you can't get a proper compartmentalization. It can break easily. Okay, and if the concentration increases, steric hindrance and all that comes into picture. So now more phospholipids come into picture. That is when your bi-layer comes. So the phospholipid assumes a bi-layer. 2 layers of phospholipid come. Okay, so what that does is the hydrophilic part on one side will attach itself to, attach as in it is more in in contact with that. So that will be towards the extracellular fluid. The other part is going to be inside, just like we saw in the micelle. But inside also we need water. For the cell, the intracellular fluid should be there. So again, the other part is going to go inside. The hydrophilic part will be inside and the hydrophobic part will be between them. So there will be a sandwich layer of hydrophobic part, and on the ends, one hydrophilic part to the extracellular fluid, one hydrophilic part to the intracellular fluid. That is your basic structure of your plasma membrane. Now, this plasma membrane can assume a lot of shapes. I want you to forget everything you know about plasma membrane. It's not as simple, and plain as you would imagine it to be, or how you were taught. Okay, so I'll tell you a few things that will pique your interest. Your plasma membrane is a very dynamic structure. So what do I mean by that? at every point within the plasma membrane, it's all different. So what happens is, it all depends on the composition. What type of phospholipid is there? Apart from phospholipid, what else are there? What is the protein structure that is there? Some proteins get lipidated and all that. Now there is cholesterol presence. So the presence of cholesterol. How much is the interaction between the phospholipid? And you should know that lipid, the phospholipid, is actually asymmetrical. You have two leaflets, one on top and one below. These two leaflets are not the same. The phospholipids that are there are completely different in each of these leaflets. So that also changes, and they can flip amongst themselves. Now, apart from all this, the temperature, the length of the fatty acid chain, the double bonds that is there, everything affects your plasma membrane. And if you take a whole length of a plasma membrane, the structural differences between each layer, each zone is completely different. So that is what microdomain is. So your plasma membrane is further divided into microdomains, and each of these microdomains have their own changing different fluidity, permeability, and even shape for that matter. So that is why, because of the association with your glycocalyx outside and cytochemical structures inside, it can help in microtubule formation and movement of pseudopod, tubular shapes, lot of things. So your plasma membrane is an ever-changing dynamic structure which has lipid asymmetry, which not only has phospholipids, it's not pure phospholipid layer. In eukaryotes, it's a little more complex. Okay, it has proteins, cholesterol, all these other structures are there, and even carbohydrate. Okay, so that is your plasma membrane. So once you understand this concept, let's go on to understand what are the structures of phospholipid. Now when you talk about phospholipid, so this most common phospholipid that is there is phosphoglyceride. So there is a glycerol component there. So one part of the glycerol is there. Now to this glycerol, two fatty acid chains are there. Okay, so to glycerol, one fatty acid, two fatty acid, three fatty acids are there, that becomes a triglyceride. But this is not a triglyceride structure. So there's one fatty acid, another fatty acid chain. And to one end of it will be phosphate. So you need something hydrophilic. So phosphate is there, and to that phosphate head, something else is attached. So what is attached there determines what type of phospholipid it is. So there can be inositide, there can be choline, there can be serine, ethanolamine. So all that will determine what type of phospholipid it is. But remember, all that we talked about right now is glycerol based. So they're all phosphoglyceride, whatever I mentioned so far. Now replace this glycerol base and put in sphingosine. Now we have an entirely different type. So now we have sphingosine based lipids. Okay, so one example is your sphingomyelin. You can see over here the sphingosine is coming instead of glycerin. Okay, now you can get it a little more complicated by changing the head over there. So galactose coming into picture, so galactocerebroside, now more oligosaccharide, the saccharide units are changing over there, ceramide and all that, so gangliosides are coming. So whatever it is, they're all sphingosine based. Now completely apart from this, you have your cholesterol. Okay, so cholesterol has its own function in your plasma membrane. We'll talk about it in a separate lecture. Now once this is done, I want you to understand that something called lipid asymmetry is there. So all these things that we talked about just now, in each layer, the one that is there outside, that is the outer leaflet, then inside is the inner leaflet. The outer leaflet and the inner leaflet, the composition of your phospholipid is completely different. Okay. Now inside phosphatidyl, inositol, ethanolamine, serine, all these are there inside most commonly. Sphingomyelin, phosphatidylcholine, these are there out in the outer leaflet. So this sort of lipid asymmetry is there. Now, can hydrophilic things move through the membrane? Normally, in a pure phospholipid layer, it cannot move. Okay. Now, this phospholipid, as we know it, the fatty acid length and all that determine a lot of things in it. So there is some sort of movement which is possible, a lateral diffusion. The leaflets can move, it can rotate, it can flex, the heads can move. Okay, so this sort of movement, controls your fluidity. It's all based on the temperature initially. Okay, and as I mentioned earlier, the double bonds and the length of the fatty acid chain. Okay, so again about fluidity and all that, I'll put a separate video out just to describe that. So I hope you understood this. Now, as I was talking about lipid asymmetry, the interesting thing about this, there is functional importance to all this. There's a reason why these things are there outside, these things are there inside. There are functional importance for all this. Phosphatidylinositol, which is the inside, it is a very important signaling mechanism that gets split into IP3 DAG to give signaling cascades, protein kinase C. So that is one thing. Now this thing gets flipped. Usually from high concentration it goes to low concentration. So this is a very slow mechanism. So in our body there are enzymatic reactions that take place. Flippase, floppase, scramblase, all that can change. So this again in detail I'll put up another video. Okay, so I hope you understand what a basic phospholipid layer is, what it does and how it is. Now once you understand this picture and you have a clear concept of what a phospholipid layer is, then we can go forward. Okay, so thank you.